Okay, so for this one, we're going to look at B cell or B lymphocyte activation here. You have to remember you're going to need some stuff from the T lymphocyte activation. And I also want you to remember we're in the lymph node here. B cells and T cells that are mature are in the lymph node together. Our virus has entered, okay, and our antigen presenting cell has eaten it, presented it to our T cells already. Now we're going to see how are we going to activate the B cells in the same lymph node. Okay, so B cell or B lymphocyte activation is going to be just a little bit different than T cells. And again, I wanted you to see how in your lymph node, you're going to have multiple B cells, and each of those B cells are going to have receptors, but in this case, the receptors are actually going to be antibodies, okay? And because you have such a large population, what you essentially should see is that these antibodies have their own specific variable region. Ooh, that was really close. Sorry, I'll do this one more time in a different way. Is that you're going to have a large population of B cells, and they all have their own like kind of different structure for their variable region. Okay, that's what's happening here. So what's going to happen is the B cell is going to basically, once it binds to an antigen that's going to be on one of our pathogens here, this is our virus. The goal was so I didn't have to draw this, but that is in a weird place. Anyway, besides the point. So our B cell has these antibodies with different variable regions here. And what's going to happen is as soon as the antibody attaches to the antigen on the surface of our virus or our pathogen, then we're going to bring this into the cell itself. Okay, so just imagine we engulf that. And then what we're going to do is we're going to push it out, essentially here, and represent a portion of that pathogen on the surface, okay? So the B lymphocyte brings in the pathogen and then represents a piece of it on its surface. When it does this, it acts kind of like an antigen presenting cell. And what ends up happening is you'll have, ooh, what color did I use? The lighter blue. Let's use the lighter blue. You'll have an activated helper T cell that now has been activated for that same antigen. So the activated helper T cell is going to come to the area here, and what it's going to do is bind to the antigen. It double binds here, but anyway, what it's going to do is it's going to release cytokines. And when it releases cytokines, now our B cell is activated. And that's going to cause our B cell to start to divide and proliferate. Like really specialize and proliferate, okay? What you're going to end up with is some memory B cells. And again, these memory B cells on the surface is going to have antibodies with a variable region that's not variable anymore, that is going to have single specificity for that antigen that it got activated by, okay? You're also going to create plasma cells here. What you should notice is the plasma cells don't have any antibodies on their surface. Instead, what they're going to do is they're going to make antibodies, and then they're going to release those antibodies. Okay. I'm just going to draw one. Sorry. It takes a long time to draw all this stuff. Because I should just animate it. But so we're going to make and release antibodies. Okay, so this is why this is fluid response, because now all these antibodies are going to be made and released in the lymph, and they're going to circulate all around the body. So when they encounter another virus here with all these spike proteins on the surface, essentially now what we can do is we can bind here and neutralize. Like maybe you have another virus over here that you're also neutralizing at the same time, okay? And antibodies have multiple responses. Mostly what they do is they're going to bind to the antigen. And they're going to cause things like neutralization. They cause agglutination, precipitation, but it's all kind of neutralization, okay? They can then, once something's neutralized, trigger phagocytosis, which remember is just eating. So now that all these viruses are bound to antibodies, a macrophage will come in and eat it, okay? And then they can also trigger the complement system. Essentially, again, in that case, the bacteria explodes. Those are like the three like main things that are important. It does also trigger inflammation as well, but sometimes that's like a negative, like not a great effect. Like if you have allergies and things like that. Okay. So this is B lymphocyte activation. Hopefully it's not too bad. So we're going to use the memory cells here, and we're going to use the memory cells that we created from T cell or T lymphocyte activation to think about vaccines a little bit more, okay? So I have this chart in here that I'm going to leave blank as a practice for you guys. Just imagine if you have SARS-CoV-2. That's actually the virus that caused COVID-19. I want you to think about like how are you going to create a flow chart for activation of B cells and T cells, okay? Just as a practice for you. Is this something that would be on the test? It's maybe, but you know, like, yeah, maybe. There could also be multiple choice questions about it.